we introduced a framework for assessing cell-to-cell expression variation, or CEV in short, and its association with phenotypes or genotypes in humans. To illustrate the concept, panel A shows a population of cells expressing protein X at different levels, where darker blue represents higher expression and lighter blue for lower. Such CEV has been shown to occur naturally and be regulated by cells, but its functional significance, particularly in humans, is not well understood. To quantify the degree of CEV for protein X in this cell population, we use a statistical metric such as the standard deviation to capture the variation of expression levels among single cells. In contrast to panel A, where the cell-to-cell differences span a continuum, panel B shows another population where the cells form two discrete subsets based on their expression level of protein X. In this particular case, the cell population has a higher CEV than the previous example. To systematically assess CEV, we define heterogeneity parameters, or HPs in short, for combinations of protein, cell population, and the statistical metric used. One example HP is the standard deviation of HLA-DR in CD4 positive T cells. It's important to know that the same HP can have different values from one subject to another, and within the same subject, it can also change over time. Panel D shows the workflow of our framework, developed by using data from a healthy cohort. There, from 61 subjects, we collected PBMCs at 3 baseline time points spanning 2 months and assayed each sample using multicolor flow cytometry. In every subject and time point, we calculated the HPs corresponding to different combinations of the 14 proteins measured and the 28 cell populations gated. Our analyses show that over 70% of the HPs were relatively stable over 2 months within a person, but exhibited substantial person-to-person differences. A number of CEVs were found to associate with either age or disease-related genetic variants. On our website, you can inspect full details of the results through interactive figures. For example, heterogeneity of CD25 and CD27 positive T helper cells tended to decrease with age. In the center plot, the x-axis is age, the y-axis is the HP, and each dot is a subject. We can interactively select any of the subjects to inspect the underlying single-cell distributions on the right. We can do the same for genetic associated HPs, such as the heterogeneity of CD38 in naive T helper cells, which was found to associate with an asthma-related SNP. Here, the x-axis is the copy of the minor allele, and the y-axis is the HP, and each dot again is a subject. In sum, our results suggest that CEV of peripheral immune cells is a new class of human phenotypes, potentially useful for immune monitoring, and CEVs may play functional roles in aging and contribute to disease susceptibility. Our conceptual framework and computational approaches can be applied to other flow cytometry data to study CEV. The data and software can be obtained from our website.